Hey guys, welcome back to another episode of my channel. My name is Keika, and we're going over concept 5.6. Genomics and proteomics have transformed biological inquiry and applications. Experimental work in the first half of the 20th century established the role of DNA as the carrier of genetic information, passed from generation to generation, that specified the functioning of living cells and organisms. Once the structure of the DNA molecule was described in 1953, and the linear sequence of nucleotide bases was understood to specify the amino acid sequence of proteins, biologists sought to decode genes by learning their nucleotide sequences, often called base sequences. The first chemical techniques for DNA sequencing, or determining the sequence of nucleotides along a DNA strand, one by one, were developed in the 1970s. Researchers began to study gene sequences, gene by gene, and the more they learned, the more questions they had. How was expression of genes regulated? Genes and their protein products clearly interacted with each other, but how? What was the function, if any, of the DNA that is not part of genes? To fully understand the genetic functioning of a living organism, the entire sequence of the full complement of DNA, the organism's genome, would be most enlightening. In spite of the apparent impracticality of this idea, in the late 1980s, several prominent biologists put forth an audacious proposal to launch a project that would sequence the entire human genome, all 3 billion bases of it. This endeavor began in 1990 and was effectively completed in the early 2000s. An unplanned but profound side benefit of this project, the Human Genome Project, was the rapid development of faster and less expensive methods of sequencing. This trend has continued. The cost for sequencing 1 million bases in 2001, well over $5,000, has decreased to less than 2 cents in 2017. And a human genome, the first of which took over 10 years to sequence, could be completed at today's pace in a day or less. The number of genomes that have been fully sequenced has exploded, generating reams of data and prompting development of bioinformatics, the use of computer software and other computational tools that can handle and analyze these large data sets. Just a question that came into mind. We're talking about bioinformatics, and we live in the day and age of the rise of AI. Do you think scientists would trust bioinformatics and AI together? Just a question. Let me know in the comments what you guys think. The reverberations of these developments have transformed the study of biology and related fields. Biologists often look at problems by analyzing large sets of genes or even comparing whole genomes of different species, an approach called genomics. A similar analysis of large sets of proteins, include their sequences, is called proteomics. Protein sequences can be determined either by using biochemical techniques or by translating the DNA sequences that code for them. These approaches permeate all fields of biology, some examples of which are shown in Figure 526. So, contributions to genomics and proteomics to biology. So, we gave rise to evolution, conservation biology, species interactions, medical science, and paleontology. And paleontology is the study of our extinct relatives. The study of the DNA, and I guess the comparison of our DNA and ancient DNA. Perhaps the most significant impact of genomics and proteomics on the field of biology as a whole has been their contributions to our understanding of evolution. In addition to confirming evidence for evolution from the study of fossils and characteristics of currently existing species, genomics have helped us tease out relationships among different groups of organisms that had not yet been resolved by previous types of evidence, and thus infer evolutionary history. Okay, so for my notes, I wrote, once the structure of a DNA molecule was described in 1953, the linear sequence of nucleotide bases was understood to specify the amino acid sequence of proteins, biologists sought to decode genes by learning their nucleotide sequences. So that's basically the first sentence. To fully understand the genetic functioning of a living organism, the entire sequence of the full complement of DNA, the organism's genome, would help enlighten biologists. Biologists started a project in the 1980s to sequence the entire human genome, and completed in the early 2000s. The Human Genome Project resulted in faster and less expensive methods of sequencing. Over 5,000 in 2001, less than 2 cents in 2017. One genome took 10 to sequence. Now, 10 took... Hold on, let me just... One genome took 10 years to sequence. Now it's done in a day or less. Bioinformatics uses computer software or other tools that can handle and analyze large data sets of genome sequences. Genomics. Solving problems by analyzing large sets of genes or even comparing whole genomes of different species. Proteomics. Analysis of large sets of protein, including sequences. Genomics and proteomics contribute to the understanding of evolution through the relationships between different groups of organisms. Yes. Okay. DNA and proteins as tape measures of evolution. Evolution. We are accustomed to thinking of shared traits such as hair and milk production in mammals as evidence of shared ancestry. Because DNA carries heritable information in the form of genes, sequences of genes and their protein products document the hereditary background of an organism. The linear sequences of nucleotides in DNA molecules are passed from parent to offspring. These sequences determine the amino acid sequences of proteins. As a result, siblings have greater similarity in their DNA and their proteins than do unrelated individuals of the same species. Given our evolutionary view of life, we can extend this concept of molecular genealogy to relationships between species. We would expect two species that appear to be closely related based on anatomical evidence, and possibly fossil evidence, to also share a greater proportion of their DNA and protein sequences than do less closely related species. In fact, that is the case. An example is the comparison of the beta polypeptide chain of human hemoglobin with the corresponding hemoglobin polypeptide in other vertebrates. In this chain of 146 amino acids, humans and gorillas differ in just one amino acid, while humans and frogs, more distantly related, differ in 67 amino acids. In the scientific skills exercise, you can apply this sort of reasoning to additional species, and this conclusion holds true as well when comparing whole genomes. The human genome is 95 to 98% identical to that of a chimpanzee, but only roughly 85 identical to that of a mouse, a more distant evolutionary relative. Molecular biology has added a new tape measure to the toolkit biologists use to assess evolutionary kinship. Human, rhesus monkey, and gibbon. So we're gonna skip these exercises. Oh, and we're finished. Okay. So we're gonna go over my notes. I need to drop my pen. It's okay. Since DNA carries heritable information in the form of genes, sequences of genes and their protein products document the hereditary background of an organism. Molecular genealogy. Molecular genealogy. That two species that appear to be closely related based on anatomical evidence share a greater proportion of their DNA and protein sequences than do less closely related species. The human genome is 95 to 98 identical to that of a chimpanzee and 85 to a mouse. Molecular biology helps biologists assess evolutionary kinship. Okay, that is it for this section and the whole of chapter 5. I finally finished chapter 5, you guys. So next video we'll be going over unit 2, the cell, and chapter 6, a tour of the cell. Okay, so I'll see you guys next time, and let me know what you guys think in the comment section below. Have a good one. Bye bye.